a video on the fungi. The study of fungi is called mycology. And if this video interests you, we actually have an entire course of mycology here at UVU that you're welcome to take. As we look at mycology, we're going to talk about the different types of fungi, mainly yeasts and molds. We're going to compare their structures, and we're going to talk about their role in the environment. Okay, let's go ahead and get started. Fungi consist of yeast, molds, and mushrooms. But in this class, we're only going to focus on the yeasts and the molds, as those are the microscopic fungi, while mushrooms are macroscopic because we can see them. Interestingly, fungi are the closest living organisms to animals. If you look back at the tree of life, you'll see that the animal branch and the fungi branch are relatively close together. The majority of fungi are what we call saprobes, meaning that they secrete enzymes out into their environment. So if we have a fungal cell, that fungal cell is going to produce digestive enzymes inside. It's going to secrete those out into the environment, which are then going to take molecules such as carbohydrates or proteins and digest them into the individual subunits, which are then reabsorbed into the cell to be broken down and used for energy or to make their own cellular structures. This is why fungi are able to play a key role in the carbon cycle of the ecosystem. They are one of the primary decomposers, with their digestive enzymes helping to break down the macromolecules of dead organisms and recycling those molecules back into the environment. One concept that is sometimes hard for students is that you are also a saprobe. So your cells produce digestive enzymes that are secreted into your intestines, where they digest your food, and you then absorb that food back into the body. Although the intestine is housed within the body cavity, it's technically a hollow tube extending from mouth to anus, making it external to the internal intestinal epithelium. The benefit for organisms having a body cavity is that we've trapped our digestive enzymes and digested food in one place for reabsorption, while fungi risk those digestive enzymes or food diffusing away from it instead of being reabsorbed into the cell itself. While fungi are similar to animals, they do have some key differences. The two biggest being that they do contain a cell wall, and that cell wall is composed of something called chitin, which is different than plant cell walls, which are made of cellulose. Chitin is actually similar to what makes up insect shells. Additionally, their plasma membranes have something called ergosterol, which is shown here in the image. Ergosterol is very similar to cholesterol in animal membranes. These differences become important for pathogenic fungi because differences between pathogens and our cells make important targets for drug treatments. There are some pathogenic fungi that cause diseases called mycoses, but the majority of them are what we call opportunistic pathogens. These do not cause disease in people with a healthy immune system, but instead use a weakened immune system as an opportunity to invade the body. So most fungal diseases occur in people whose immune system has been weakened from some other cause. Microscopic fungi can be classified as mold or yeast based on their morphology. Molds you are probably familiar with as the fuzzy stuff that you've seen growing on bread. These fuzzy things are hyphae structures of mold. Hyphae are long chains of cells that are either separated by cell walls, seen here called septate hyphae, or non-septate hyphae, which you'll notice have no cell walls and are just one long cell with multinuclei. The benefit of hyphae is that they extend through the environment, increasing the surface area available to absorb digested nutrients. The ends of the hyphae will form structures that hold spores of the mold. This is often what you see at the macroscopic level on bread. Interestingly, some molds have actually been very beneficial for humans because they produce many of the antibiotics that we use to treat bacterial infections. They're also used in the food industry, especially in cheese production. If you've ever had or seen blue cheese, the blue you observe is actually the mold Penicillium roqueforti, which gives blue cheese its distinctive taste. Molds will switch back and forth between asexual growth and sexual reproduction when nutrients get depleted. In asexual growth, the hyphae are going to grow longer break off every once in a while, where they will then spread and continue growing hyphae. Groups of hyphae are called myceliums. However, under certain conditions, the mycelium can actually join together in fertilization, sharing their genetics together and then forming gametes. This occurs through meiosis. These sexual spores are then released into the environment where they find some place to land, germinate, and then start developing hyphae again, returning to the asexual cycle of growth. It is the aerosolized spores during sexual cycle that often cause medical problems for people. This is what you find associated with things like black mold, because those spores can germinate in the lungs if inhaled. Different species of molds have slightly different replication cycles. You do not need to differentiate the reproductive cycles of different species of molds have slightly different reproduction cycles. You do not need to differentiate the details of the different reproductive mechanisms associated with different molds for this course. The other macroscopic fungi morphology are yeast. These are small single-cellular organisms that reproduce asexually by budding. Budding is slightly different than mitosis because the cell does not divide in half after DNA replication. Instead, the new daughter cell grows out of an extension of the plasma membrane from the parent cell. So you can see in this image here. We can see one bud that is starting to form and another that is just about finished. When a bud is large enough to be its own cell, it will break off from the parent cell, leaving what's called a bud scar. A single yeast cell cannot produce another bud from an area of its plasma membrane that already has a bud scar. Thus, when the surface of the cell is completely covered with scars, it can no longer replicate. Similar to molds, haploid yeast cells are also able to fuse together through mating and sexual reproduction to form a diploid cell that will then undergo meiosis to regenerate haploid yeast cells, which will then move back to budding. Interestingly, some fungi are dimorphic, meaning that they exist both as a yeast morphology in some environments and a mold morphology in others. Until we had molecular sequencing techniques, we never actually knew that the two morphologies of dimorphic fungi were the same species. So most of these organisms actually have two completely different genus and species names, one for their mold form and one for their yeast form, even though they're actually the same organism. We'll come back to these types of fungi in a minute when we talk about pathogenic fungi. When you compare yeasts and molds, there are key morphology, reproductive, and habitat differences. When they grow on Petri plates, you can clearly see the fuzzy structures of molds that grow, but yeast actually look much more like bacterial cells growing on a Petri plate. Under an electron microscope, you can also observe the key reproductive differences, with molds forming spores and yeast forming buds. While most fungi are not pathogenic, there are a few that cause important diseases. The study of pathogenic fungi is called medical mycology. Interestingly, you find that many dimorphic fungi can be pathogenic, with the mold form growing in the environment, which then releases spores during replication, which can be inhaled by an individual. Once those spores germinate in the lungs, they actually grow in the yeast form. These allow them to easily spread throughout the body through the bloodstream and the lymph tissue. However, once they get to the tissues, they then switch back to the mold form, and the hyphae allow them to invade tissues, leading to disease. The most common fungal pathogen is Candida albicans, which is actually part of the normal flora of humans, but if the fungi enters the bloodstream or becomes growing in high numbers, then it can cause serious infection. It's one of the most common causes of nosocomial or hospital-acquired bloodstream infections that we see. Other pathogens like Aspergillus produce a toxin that causes the disease itself. Since most fungi are opportunistic pathogens, people who are HIV positive are often susceptible, with Cryptococcus neoformans being one of the leading causes of meningitis in HIV patients and in people who have undergone organ transplants because they must be on an immunosuppressants for the rest of their life.